Hi everyone, welcome to video 12 of unit 3. So we just finished up those different divisions of the nervous system, and now we want to look at these neurons, these functional units, these functional cells that are going to be carrying our nervous impulses through those different divisions of the nervous system. Okay, so this is a neuron. A neuron is a cell, and we'll say it's the functional cell of the nervous system. So it's going to conduct impulses. And you can get familiar using this word action, these words action potential instead of nervous impulses. Alright, so let's start identifying some different parts. So as we start talking about this, it makes a little bit more sense. So up here, we have these extensions from the cell body. These are going to be called dendrites. So dendrites play a role in reception. So we'll say this is the receptive field. So whether it's taking some sort of stimulus, like heat or pressure or anything like that, or temperature, and it's getting stimulated like that, or if it's another neurotransmitter that's going to be sending a message. Either way, these dendrites are going to be the thing that's going to be detecting whatever this stimuli is. So we have the dendrites. Next, we have the cell body. So the cell body can also be called the soma or the perikaryon. We'll talk about a few more things with the cell body in a minute. Then we have this whole thing over here. So this is called the axon. Oops. So this is our axon. And there's a few different parts that we want to be able to tell. Okay, so I'll do a different color. This is all going to be stuff of the axon, related to the axon. So here, this is the origin of the axon. That's called the axon hillock. Over here, this is called the telodendria. This is your end. Telodendria. I need to stop circling these things before I write it. That's going to be our telodendria. We know that these little endings here, these are called synaptic knobs or synaptic end bulbs. So I'll say knobs over here, or I can say end bulbs. Those are going to have those synaptic vesicles with neurotransmitters, so that's going to be one there and one there. This is pretty rare. This is a side branch of an axon. It's called an axon collateral, so we don't really see those too much, but I drew this one with a axon collateral just to identify one. And then we know that some of our axons have this wrapping of lipid around them. That's called the myelin sheath. We'll talk about what that's going to do for conduction a little later. So that's your myelin sheath, and that's EATH. And then the spaces in between, so I'd probably have more myelin than that, right? I'd have more myelin like this. So these spaces in between are myelin. These are going to be called nodes of Ranvier. Anything else that I want to talk about? This is lipid. We're going to talk about how it increases nervous conduction. Add some protection for the neuron. So some neurons aren't going to have a myelin sheath, so we'll have myelinated versus unmyelinated axons. Okay, so I want to do one more thing. Let's just do this really quickly. We talked about dendrites. These are going to play a role in the receptive field. Then we had the soma or cell body or perikaryon. So there's a few things we want to talk about that's located in our cell body. So we already know it has things like nucleus and organelles, mitochondria, ribosomes, those sort of things. But we've also got a few different things. So one is called chromatic substance. Also called a Nissl body. Okay, so what is it? What is our chromatic substance or Nissl body? This is going to be a specialized rough endoplasmic reticulum. I'm going to say rough ER. If you remember, the endoplasmic reticulum makes proteins, so this protein is going to be, these proteins are going to be, the proteins made by this Nissl body or this chromatic substance are going to be used for neural growth. So it's going to make growth factors. And instead of growth factors, we're just going to call these proteins. So he's going to make proteins to aid in neural growth and repair. And we'll talk about how only certain nerves in the nervous system can repair themselves. Okay, so one thing is if you damage the cell body, you can't repair it. So you have to damage the axon, a part of the axon. So we'll look at that later. Okay, so we have chromatic substance. We've also got these things called neurofibrils. So these neurofibrils are microtubules. Microtubules were a component of cytoskeleton. That was like a Unit 1, but we've also been talking about these different filaments within Unit 2 and Unit 3. So microtubule is just another type of protein. So these are going to function in nutrient transport within the neuron. So I definitely recommend if you can type in, it's called like Life of a Cell or something. It's this Harvard animation on YouTube, but it shows you all these organelles in a 3D animation working. It's not like the ones he shows in class from like the 70s. I mean, this is like a really highly detailed animation, and you can see how you'll have microtubules like this in the cell. So this is the cell, and I'll have something like glucose. And that glucose will be attached to this microtubule, and then there will be a protein moving this glucose along this microtubule. So it's not like things are just floating randomly around the cytoplasm. They go along these structured tracks. It's really cool. You should check it out if you have time. Actually, I think you should check out that video just because it helps make sense of everything that we've been talking about. Being able to actually visualize these things working within a cell because they're microscopic, I think helps solidify the information. So the soma, the cell body, has nucleus and organelles. It's got this chromatic substance, which are making these proteins to help the neuron grow. It's got these neurofibrils to help move nutrients around the neuron. We also have lipofuscin. So this is a cellular inclusion. It's a pigment. It's this yellowish brown pigment. It increases as you age. And just so you guys know, this is an arrow. Every time I do this, I don't know why it looks like that, but it does. So those are arrows whenever I do that. They never come out like that. So it increases as you age. And we don't know what it's linked to, but we know it's not linked to dementia or Alzheimer's. Okay, so we talked about the dendrites, how these were for cellular or for reception. So whether it's getting information from a stimulus outside the body or neurotransmitters, it's going to be dealing with this reception. Then if we get that action potential, that nervous impulse, it will go down our cell body and then down our axon. So I want to do the axon one more time and just name some of those things that we looked at. So this axon, this extension of the plasma membrane away from the cell body, we had the axon hillock, which was this origin of the axon. We talked about the teledendria, which is that area at the end of the axon terminal, the ending of it. We had the synaptic knobs or the synaptic end bulbs, which had the vesicles holding our neurotransmitter. We talked about axon collaterals, which were those side chains of the axon, which aren't very common. And we looked at the myelin sheath and the nodes of Ranvier. So you guys should get familiar with this picture. This one back here, be able to identify everything on here. Alright, cool. So that's an axon. That's a neuron. Alright, moving from there, we want to look at some physiological characteristics of mature neurons. So basically, like, what are these neurons doing? How are they acting? Okay, so what are these neurons doing? These neurons are conducting nervous impulses. So as they're doing that, we want to give some other characteristics about them. So one thing is that they have this idea of longevity. So mature neurons are going to function for life. And that's important because, as you see what I'm writing on the second bullet, is that they lose the ability to divide, which sucks. So we said that they're going to be stuck in G0. So it's not going to be copying its DNA. It's not going to be making duplicate copies of its organelles to be able to split. So it's stuck in G0. This G comes from interphase, so when we talk about interphase, we talk about the G1, the S phase, and the G2. So we're not in interphase because we're not going to be doing these things getting ready to divide, and we're also not in mitosis or meiosis, so we're not cellular in the process of cellular division. So we're in G0. This is also an issue because when you damage the neuron, that causes death to the neuron. If you damage a neuron bad enough, it's not going to be able to regenerate, right? So if you cause too much damage, they haven't been dividing, so as you're losing them permanently. So if you kill it forever, and then if you cause damage to the cell body, I'm not even going to put that in there. I already talked about that. So they lose the ability to divide. They're stuck in G0. They function for life, which is a good thing. But if they divide, they're gone forever. No replacing them. And then if they're damaged really badly, they may be able to be repaired depending on where they were damaged. So apparently this is not as clear as we think it is. Apparently maybe you can get certain cells to divide. So I don't know. I guess there's some research coming out about that. The other characteristics about their physiology is that they have a high metabolic rate. So these things are constantly causing action potentials, moving sodium in, moving potassium out, and then using that sodium-potassium pump to reverse that once the action potential is done, which uses ATP. The whole point is these things have a huge metabolic activity, so they need a lot of oxygen and a lot of glucose to be able to do what they do. High metabolic rate. So lots of glucose, lots of oxygen. Alright, we're going to end this with the neurons, and we'll switch over to the glial cells, which is the other 50% of the nervous system, these helper cells. So this is the anatomy of the neuron and just a couple characteristics about this.